Quickly, Michael, HK2 actinium, you've worked with this drug.
Yeah, so I think that, the biggest limitation for going to alpha PSMA is directed therapy for the small molecules is xerostomia, and also a totally new target and one which has been around for a long time but never before the contemporary era been leveraged is actinium HK2. HK2 is human kallikrein-2, which is very closely related to PSA, but is membrane bound in addition to being soluble, as unlike PSA, which is just soluble. And we are adaptively dosing this, so they get one single dose and then don't get redosed until they progress in order to avoid what otherwise would be emerging interstitial lung disease and also persistent thrombocytopenia. So I just wanted to have this slide here as an example of a new target, adaptive dosing, avoiding the dose-limiting toxicity of current small molecule-directed, PSMA-directed alpha therapy in one agent.
Yeah.
And Tanya has experience with this drug, as does Karen, and they have a maybe objective view of these data. So we'll let them speak to it.
No, I mean, the activity we've seen on trial, as those who can see the slide, is impressive. And I think the adaptive strategy is certainly what's moving us forward. Toxicity-wise, there was pneumonitis as an unexpected toxicity that seems to be getting sorted out with this adaptive dosing, but certainly something new relative to at least our experience with lutetium.
I've been a little surprised by the cytopenias, given that it's an alpha particle with a narrower range of energy deposition. But I don't know if that just speaks to the population or if you have any thoughts about.
It's an antibody, so its clearance from blood is much slower than a small molecule. Now that's a double-edged sword, so it does have a longer circulating half-life than a small molecule does. On the other hand, that may account for its really long duration of effect because the internalization and retention in the tumor is longer as well. So at least we hypothesize, which is why, like after one dose, you can see a treatment effect that's 6 months, 12 months, et cetera. So it speaks to what we started this morning out with in discussing the bispecific antibodies, is finding the right molecule that minimizes tox and maximizes treatment effect. And in radioligand therapy, a lot of that isn't just payload, but also the size of the targeting molecule. And this is a big targeting molecule. Yeah, go ahead.
I was going to say, sounds like a good example of sort of that this is just the beginning of the road, right? Sorting out target and molecule and sequential therapy, what I mean? This is the beginning of the story, not the end.
Right.
But the duration of responses is really impressive.
Yeah.
Yeah.
All right.
Because actinium is not just a single alpha, it's actually a decay cascade. Has there been biokinetics to look at the byproducts? Because if it's blood-bound longer, these secondary alphas and betas are in that chain and potentially are localizing elsewhere besides the tumor cells.
Great point, Gary.
Yeah. All right, this is the last slide, very simple slide. This is basically just to say this field is exploding, right? I mean, like I said, beginning of the story, I can't even count how many companies sort of involved. Find your favorite company in there. The field is sort of exploding, and I think we'll probably have. Tom's telling me to hurry up, boy. I mean, now I've seen everything. We're finishing, yes. I'd like to thank all the prostate cancer panel. Thanks for a great session.